Okay, so welcome to this video. In this video, what we're going to discuss is the GABA-A receptors. Okay, now the GABA-A receptors, notice the plural. There is not just one GABA-A receptor. There are a huge number of different GABA-A receptors. It's an entire family of receptors. Okay, right. Now, the GABA-A receptors are within the larger family of ligand-gated ion channels. Okay, so remember there are four main types of receptors. There are the G protein coupled receptors, the ligand-gated ion channels, of which the GABA-A receptors are examples, the enzyme coupled receptors, of which the biggest and most notable family is the tyrosine kinase receptors, okay, and then finally the nuclear receptors, such as the receptors for steroids, okay, which then change gene expression in response to their ligand binding. Okay, right, so the GABA-A receptors are all ligand-gated ion channels, and what this means is that they are an ion channel that is usually closed, okay, but when the ligand binds to it, it will cause that ion channel to open, and then ions will be able to move through that ion channel, and that will change the electrical potential difference across cell membranes. Okay, so ligand-gated ion channels are often abbreviated to LGICs for short. So L for ligand, G for gated, I for ion, and then C for channel. Okay, so ligand-gated ion channels. Okay, right. So, how are we going to structure this video then? We're going to start off by discussing the ligand itself. We're going to start off by discussing GABA. Okay, then what we'll move on to is the classes of ligand-gated ion channels, because there are a huge number of different ligand-gated ion channels in the human body, and we characterize them into three main families. So I'm going to talk about those three main families. We'll see which family the GABA-A receptors fall into. Then we'll have an in-detail study of the structure of GABA-A receptors, and then we'll look at the function of GABA-A receptors, so what happens when ligand binds to them, okay, how that's going to affect the electrical potential difference across the cell membrane, and then we'll put it into the bigger picture of whole neurons, basically, and what effect it's going to have on the likelihood that a neuron fires an action potential. Okay, right. So, the ligand then for GABA-A receptors is GABA itself. Okay, now GABA stands for, okay, Gamma-Aminobutyric Acid. So the G is for gamma, okay, the A is then for amino, and the B is for butyric, and then the A is for acid. So Gamma-Aminobutyric Acid. Okay, so G is for gamma. Okay, and often people will just abbreviate gamma down to the Greek symbol. Okay, so they'll abbreviate this to gamma in the Greek symbol, like so. Then amino is the A, butyric is the B here, and then A is for acid. Okay, right. Now, let me show you the structure of gamma-aminobutyric acid, and then we'll talk about a subtle point, okay, concerning gamma-aminobutyric acid as it exists within the normal extracellular fluid. Okay, so let me draw it then. So butyric acid, let's start off with what butyric acid here means. Butyric acid is the old name for a molecule that would now be called butanoic acid. Okay, so this is what would now be called butanoic acid, and that means a 4-carbon fully saturated carboxylic acid. Okay, then gamma, okay, well actually I think I'll start by drawing butyric acid or butanoic acid, and then I'll discuss what gamma-amino means. So here is the carboxylic acid group. Okay, then we're going to need 3 additional carbons to take the total number of carbons up to 4. Okay, right. Now if we're just drawing normal butanoic acid, we would put hydrogens coming off all of the remaining spots that we can have bonds coming off. Okay, like so. Now, I'm going to leave one space here Okay, if we were just drawing butyric acid, there would be another hydrogen off here, but we want gamma-aminobutyric acid. Okay, so this means we're going to have an amino group coming off gamma carbon. So what does gamma carbon mean? Well, this is the old way of naming the carbons within a carboxylic acid. Nowadays, we would call this carbon number 1, this carbon number 2, this carbon number 3, and this carbon number 4, okay? However, the old way of naming the carbons within a carboxylic acid is to call the first carbon that comes off the carboxylic acid carbon the alpha carbon. Okay, so this one is the alpha carbon here. Okay, then the second carbon that comes off that we'll call the beta carbon, and then the third one we'll call the gamma carbon. Okay, so this tells you how far away your carbon is from the carboxylic acid carbon. Okay, so gamma-aminobutyric acid then means butyric acid with an amino group coming off the gamma carbon here. So all we need to do is put an amino group there, and that produces this gamma-aminobutyric acid. Okay, now, the subtle point that I want to talk about is that the actual molecule, if you put this in normal physiological pH, okay, which is around 7.45, the molecule doesn't exist in this form, okay, because this carboxylic acid group here is an acid group. Okay, so what that means is that it is capable of donating a proton away into solution. Specifically, this proton on the alcohol group here can be donated away into solution. So if we draw this alcohol group in a little bit more detail here. Okay, so I'm just drawing this in more detail. We've got the oxygen atom bound to the hydrogen atom. Okay, and in this single covalent bond we have these 2 electrons. One from the oxygen here in red, and then another from the hydrogen, which we'll have here in blue. Okay, now what can happen is the hydrogen atom here can cleave off from the oxygen atom, leaving its electron that it's put into this single covalent bond behind on the oxygen. Okay, so that will leave the oxygen atom with a negative charge because it's gained an electron, and then the hydrogen atom, now without its electron, will go off into solution. Now, what does a hydrogen atom actually consist of? Well, a hydrogen atom is the most basic of all the elements. Okay, it just consists of usually a proton with an electron orbiting it. Okay, so no neutrons is the normal form of hydrogen. There are other isotopes of hydrogen where you do have neutrons in the center, but the normal isotope of hydrogen has no neutrons in the nucleus. Okay, it just has a single proton there in green with an electron orbiting around it. Okay, and if you want the fancy name, the specific name for this isotope of hydrogen is protium, and people will call it H1. Okay, there are also other forms of hydrogen where you have, for instance, 1 neutron in the center. That's called deuterium. Okay, in that case you would call that H2 because you've got 2 nuclear particles, and then if you have 2 neutrons in the nucleus and therefore 3 nuclear particles overall, that's called tritium, and that's H3. However, the most normal form for hydrogen is the protium form, where you have only 1 nuclear particle and it's a single proton, and then you have no neutrons. So, if we assume that this hydrogen atom is a protium form of hydrogen, then once it's left its electron on this oxygen, then the proton will be cleaving off, and it will then be donated into solution, basically. Okay, so that's how this molecule can donate protons into solution, and hence it is an acid molecule. Remember, the definition of an acid molecule is one that can donate a proton into solution. Now, usually under physiological pH of 7.45, this carboxylic acid group does donate its proton into solution, and therefore what the molecule will usually actually exist as is the form where you don't have this proton here, and instead you have an oxygen with a negative charge. Now, that has another name. Okay, the molecule where you don't have the proton on here, but you have a negative charge on that oxygen atom, is called Gamma-Aminobutyric. Okay, so I'll now use the gamma symbol rather than writing gamma. Okay, so Gamma-Aminobutyric. And this is what's known as the conjugate base of Gamma-Aminobutyric acid. Okay, so this is a general concept that applies for all acid molecules. Basically, once an acid molecule has actually donated its proton away into solution, it's no longer an acid anymore. Remember, the definition of an acid molecule here was that it was a molecule capable of donating a proton away into solution. Okay, this molecule was capable of donating a proton away into solution, and therefore it was an acid. However, the molecule that we've now got, where we no longer have that proton there and just have the oxygen with the negative charge, is no longer capable of donating a proton away into solution. So it is no longer an acid, basically. Instead, it's actually a base because it can accept a proton out of solution. Protons can come and bind to this oxygen with a negative charge. Okay, and that makes it a base because the definition of a base is a molecule that can accept a proton out of solution. Okay, which indeed Gamma-Aminobutyric can. Okay, now because of its relation to the original acid molecule, because it's kind of like the other side of the coin, if you like, it's called the conjugate base of the acid, basically. And this is a running theme for all acids, not just Gamma-Aminobutyric acid. For all acids, once they've donated their proton away, the structure that is then left over is no longer an acid. It can't donate another proton away. Instead, it's actually a base because it can receive the proton back again. Okay, and it's called the conjugate base of the acid. So, usually, if you put gamma-aminobutyric acid molecules, GABA molecules, into a physiological pH solution, okay, usually they give away their protons and therefore exist in this form, gamma-aminobutyrate, rather than the actual gamma-aminobutyric acid. Okay, so that's just a subtle point that really the ligand for these GABA receptors isn't actually GABA, but it's the conjugate base of GABA, gamma-aminobutyrate. Okay, so that's just a subtle point there. Right, okay, so we've now talked about the ligand, which is this 4-carbon molecule with the amino group and that carboxylic acid group, which is usually existing in its conjugate base form. Now what we want to talk about is the different types of ligand-gated ion channels. Okay, so that we have a broader picture of where the GABAA receptors fit in this field of ligand-gated ion channels. Excuse me. Okay, right, so there are 3 main families of ligand-gated ion channels. Okay, so we'll start off with family 1. Okay, and I want to give a little bit of a discussion for each of them so that you've got a broad picture. Okay, so family number 1 of ligand-gated ion channels, and I should really call these superfamilies because each one of these 3 families is going to contain multiple different families of ligand-gated ion channels. Okay, so the GABA-A receptors, for instance, are an entire family of receptors, and they are going to be located in one of these superfamilies of receptors. Okay, so this first superfamily of ligand-gated ion channels are what are known as the P2X-like receptors. Okay, and this family, or this superfamily rather, is named after its most important members, basically, which are the family of P2X receptors. Okay, so let me show you the characteristic features that you have to have in order to be considered a P2X-like receptor. So let these 2 parallel lines here represent the inner and outer leaflet of the phospholipid bilayer. And then sitting in the cell membrane, we're then going to have our P2X-like receptor here. Okay, so whenever drawing a ligand-gated ion channel, it's a good idea to start off by drawing a cylinder. Okay, now, this is our ligand-gated ion channel sitting in the cell membrane. Now, in the case of P2X-like ligand-gated ion channels, the structure of this is going to be made up of 3 separate subunits. Okay, 1, 2, 3. So the ligand-gated ion channel is not just one protein, it's multiple polypeptides stuck together. So this one is a polypeptide, this one is a polypeptide, and this one is a polypeptide. This is the case for all of the ligand-gated ion channels which are grouped into this superfamily of P2X-like receptors, that the overall channel is a trimer of 4 separate polypeptides. Okay, so this is a trimer. Now, what we're going to do also is we're going to pull out one of these subunits that makes up a third of the full ligand-gated ion channel, and we're going to have a look at its membrane-spanning topology, because for all of the different P2X-like receptors, you're going to have a conserved membrane-spanning topology for one of the subunits. Okay, so here is the cell membrane here. Okay, and now I'll show you the structure of the polypeptide, or rather the membrane-spanning topology of the polypeptide that makes up one of these subunits of the P2X-like ligand-gated ion channel. Okay, so it's a polypeptide, so here is its amino terminus. Then this line will represent a polymer of amino acids, so it represents amino acid after amino acid after amino acid after amino acid. Then you have a membrane-spanning alpha helix which will cross the membrane. And by the way, this is the extracellular fluid side of the membrane, or the ECF side, whilst this is the cytoplasmic side of the membrane. Okay, so the polypeptide has spanned the membrane. It started off with its amino terminus in the cytoplasm. It spanned the membrane, then it had a extracellular loop like so. Then it has a second membrane-spanning alpha helix, and then on the intracellular side, you then have the C-terminus of the polypeptide. Okay, so this is the structure of an individual subunit of one of these P2X-like receptors. It has these 2 membrane-spanning alpha helices with an extracellular loop, with the amino terminus and the carboxylic acid terminus both intracellular. Okay, right, so you create 3 polypeptides that will all have this same membrane-spanning topology, and you put them all together to make a full ligand-gated ion channel in the case of all of these P2X-like receptors. So, what are the main families of ligand-gated ion channels which fall into this superfamily of ligand-gated ion channels? Well, clearly the P2X receptors are one of the families of ligand-gated ion channels within this superfamily. Okay, so there is not just one P2X receptor, there are multiple P2X receptors. Okay, it's a whole family of receptors, just like the GABA-A receptors is a whole family of receptors. However, all of the subunits of P2X receptors have this same membrane-spanning topology, and the entire ligand-gated ion channel will be made out of 3 of these subunits with this membrane-spanning topology being aggregated together. Now, the P2X receptors overall are receptors for ATP, receptors for ADP, UDP, etc. Okay, so they're receptors for nucleotides, basically. Okay, right. There is another family of ligand-gated ion channels which falls into this family of ligand-gated ion channels, which are the ASICs. Okay, and this stands for acid sensing ion channels. Okay, so the A is for acid, the S is for sensing, the I is for ion, and then the C is for channels. Okay, so this once again is a whole family of receptors, and they are trimers of 3 subunits, and each of the subunits has this membrane-spanning topology. Okay, and what are they going to be gated by? Well, they're going to be gated by extracellular protons. Okay, so a high concentration of protons extracellularly will trigger the opening of these acid-sensing ion channels, or these ASICs. Okay, so again, that's not just one receptor, there's a whole family of acid-sensing ion channels, but they all conform to this same basic structure that we've seen here. Okay, so those are the P2X-like receptors. Okay, let's go to the second superfamily of ligand-gated ion channels. So the second superfamily of ligand-gated ion channels are the glutamate-like receptors. Okay, so these are the ligand-gated ion channels for glutamate. Okay, so all of the ionotropic glutamate receptors are within this family of glutamate-like receptors. So glutamate is one of the most important excitatory neurotransmitters within the brain. In fact, it is the most important excitatory neurotransmitter within the brain. And the way that it works to produce fast neurotransmission is through ionotropic glutamate receptors, which are ligand-gated ion channels, which cause changes in the electrical potential difference across the postsynaptic cell. Okay, and they're all within the second family of ligand-gated ion channels. Okay, so let's once again draw a picture for these. Okay, so once again, this is the cell membrane here, the inner and outer leaflet of the cell membrane. Then we have our ligand-gated ion channel sitting within the cell membrane here. Okay, here is the pore going down the middle, and the whole ligand-gated ion channel now is going to consist of 4 separate subunits. Okay, 1, 2, 3, and 4. Okay, right. So all of the ligand-gated ion channels which are in this second family of ligand-gated ion channels, known as the glutamate-like receptors, are going to be tetramers. Okay, now, same protocol. What we want to do is pull out an individual one of these polypeptides, which is going to make up a quarter of the full ligand-gated ion channel, and have a look at its membrane-spanning topology. Okay, so once again, here is the cell membrane. This side will be the extracellular fluid, this side the cytoplasm. Okay, and this time the amino terminus will be extracellularly. Okay, so the amino terminus of the polypeptide is extracellularly, then it's going to come down like so, it's going to span the membrane once like so, then it's going to have this portion where you try to go back across the membrane but fail, and then you come back into the cell. Now this is known as a reentrant loop. Okay, so when you try to span the membrane, fail, and then come back into the cell, it's called a reentrant loop. So I'll just colour that portion in. So this portion in pink here is known as the reentrant loop. Okay, then after the reentrant loop, you're going to have your next membrane spanning alpha helix where you'll leave the cell. Okay, then you're going to have a loop on the extracellular side, and then another membrane spanning alpha helix coming back into the cell, and then your carboxylic acid terminus inside the cell. Okay, right, so this is the structure of one of the subunits that makes up a quarter of one of these glutamate-like receptors. Okay, right, so I just would like to give you a little bit of terminology associated with the different portions here. Okay, so firstly, this first membrane spanning alpha helix we have here, which I'll colour in red. This is known as M1, okay, so membrane region 1. Okay, then this reentrant loop, this is called the membrane region 2, so it's another region that is involved in trying to get across the membrane, so it's called M2. The second full membrane spanning alpha helix, again here in red, is called M3. Okay, and then finally the fourth membrane spanning alpha helix, the third membrane spanning alpha helix, but the fourth membrane domain is called M4. Okay, right. Something else that's important about these subunits of glutamate-like receptors is that this loop in between M3 and M4 is known as the M3-M4 loop. Okay, and a portion of this M3-M4 loop, which I'll colour in orange here, is going to interact with a portion of this pre-M1 region here, also shown in orange. And these two portions together, these are going to make up the ligand binding domain, okay, which for lack of space I'll just abbreviate to LBD, but that stands for ligand binding domain. So this is where these subunits of glutamate-like receptors are going to bind to their ligand. Okay, right. So there are the main examples of receptors which fall into this family of ligand-gated ion channels are the ionotropic glutamate receptors, and those are often abbreviated to the IGLURs. Okay, so the little I stands for ionotropic, and ionotropic just means a receptor that is going to change the permeability of the membrane to ions, basically. So it's just another name for ligand-gated ion channels. You can call ligand-gated ion channels ionotropic receptors. Okay, so these are ionotropic glutamate receptors. So the GLU is for glutamate, the R is then for receptors. Okay, right. Now, there are different categories, there are different families of ionotropic glutamate receptors, and the three main categories of ionotropic glutamate receptors are the AMPA receptors, named after an agonist for them, which is AMPA. Okay. Then you also have the NMDA receptors, okay, again named after a famous agonist for those receptors. And then finally also the kainate receptors, once again named after a famous agonist for those receptors. Okay, so there are these three families of ionotropic glutamate receptors, and all of them have the same basic structure, so they're all grouped together in this family of glutamate-like receptor ligand-gated ion channels. Okay, right, so once again I'll stress that the AMPA receptor is not just one receptor, there are multiple different types of AMPA receptors. Same for NMDA receptors, same for kainate receptors, okay? But all of them are tetramers, they consist of 4 separate polypeptides stuck together, and each of the polypeptides that makes up a quarter of the receptor has a basic membrane spanning topology that looks like so. Okay, right, so those are the characteristic features which allow us to group them all together in this great family. And obviously there's a huge difference between these ligand-gated ion channels and the ligand-gated ion channels that were in this first family, the P2X receptors and the acid-sensing ion channels. Okay, these ones have 4 subunits, these have 3 subunits. Okay, now GABA-A receptors are not in either of these families. They don't have 3 subunits, they don't have 4 subunits. They therefore are in the third great superfamily of ligand-gated ion channels, which we will turn our attention to now. Okay, so superfamily number 3. Okay, so superfamily number 3 is by far the most studied of all the families of ligand-gated ion channels. Okay, now its modern name is to call it the nicotine-like receptor family of ligand-gated ion channels. However, it has another name, it has an old name which is still in far greater use than this new name. Okay, and the old name is to call them the cis-loop ligand-gated ion channels. Okay, so this third family is the family of cis-loop ligand-gated ion channels, and this family is going to contain some very famous receptors. Okay, you might be able to guess by the new name, nicotine-like receptors, what major family of receptors is going to be within this superfamily of ligand-gated ion channels. Okay, so firstly, let's start off with the bulk structure of the ligand-gated ion channel. So, all nicotine-like receptors or cis-loop ligand-gated ion channels are going to be pentamers. So let me draw one of these here. Okay, so this is the ligand-gated ion channel sitting in the cell membrane here. Okay, then it's got the pore of the ion channel going down the middle, and then it's made up of 5 separate subunits. 1, 2, 3, 4, 5. Okay, so these ligand-gated ion channels in this final family of nicotine-like receptors or cis-loop ligand-gated ion channels, these are going to be pentamers. Okay, so they're going to be made up of 5 separate subunits bound together to make up the full ligand-gated ion channel. Okay, now what we're going to do is the same protocol. We're going to pull out a single one of these polypeptides that makes up a fifth of the whole ligand-gated ion channel, and we're going to have a look at its membrane-spanning topology. Okay, so once again we'll have our cell membrane here. Okay, and now we want to see the basic structure of the polypeptide. So, here is the amino terminus, which is going to start its life off in the extracellular fluid. Okay, then we are going to have a characteristic feature on the extracellular side, and this goes for all subunits of all types of cis-loop ligand-gated ion channels. You have a structure known as the cis-loop, extracellularly, which is this loop in the polypeptide held together by a disulfide bond between two cysteine residues. Okay, so I want to discuss this concept of a cis loop in more detail, so I'll do it down here. Okay, so once again, let this line represent a polymer of amino acids. Let it represent amino acid after amino acid after amino acid after amino acid. Now, we're suddenly going to get to an amino acid that I actually want to draw the full structure of. Okay, so this amino acid is going to be totally overrepresented. It's going to be massive compared to the others. Of course, it isn't really massive. It's just for the purpose of actually drawing its structure that it's going to look massive. Okay, so this is a cysteine residue. So we are currently at this portion here, okay, where we are forming this bond that's going to be holding the two parallel sides of this loop together. And to do that, we need cysteine residue. So here is the cysteine residue on this first strand here. Okay, so here's the amino group of this cysteine amino acid residue. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group, and then you'll go back into the polypeptide, okay, and I'm not going to draw each amino acid out, so I'm going to go back into showing them just as a line. Okay, right. Now, what's the R group of a cysteine residue? Well, the R group of a cysteine residue consists of a methylene group with a thiol group coming off it. Now, a thiol group consists of a sulfur atom bound to a hydrogen atom. Okay, so this is what's known as a thiol group. Now, it has similar chemical properties to an alcohol group, okay, because sulfur is in the same group of the periodic table as oxygen, okay, and therefore has similar chemical properties to oxygen. Now, if that was an oxygen atom there rather than a sulfur atom, you'd call that an alcohol group, okay. So, as I've said, sulfur has similar chemical properties to oxygen, so we'd expect this thiol group to have similar chemical properties to the alcohol group, and indeed it does have. Now, it's going to form a bond with another thiol group that's going to be on the cysteine residue on the opposite side, which I'll colour in pink here. Okay, so I'm going to be fortuitous and not draw the hydrogen atom coming off this sulfur atom here. But in principle, if we were drawing the pure cysteine residue, you'd have a hydrogen coming off that sulfur atom. But we're going to bind this cysteine residue to another cysteine residue via a disulfide link. So that hydrogen is going to come off, basically. Okay, then the polypeptide is going to loop around like so, and then on the other side, you're then going to have another cysteine residue. Okay, so this is now this pink residue here. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group, and then we return back into the polypeptide. Then let's put the R group here, here's the methylene group, and then we're going to have another thiol group coming off here, and then what's going to happen is both of these sulfur atoms are going to give up their hydrogen atoms, and instead of binding to hydrogen atoms, they're going to bind to each other. And it is this bond here, which is this bond here, and unfortunately I've used the same colour twice, but never mind. This is the disulfide bond which is holding this polypeptide in this loop structure here. Okay, right. So this is why it's called a cis loop, because it's a loop in the polypeptide structure that is held together by two cysteine residues. Okay, and the three-letter amino acid code for cysteine residues is CYS, just like so. Okay, so that's the origin of calling this a cis loop. Now, in between these two cysteine residues here in this loop portion of the polypeptide, you are actually going to have 13 amino acids. Okay, so let's put these in. So here's the end of this one, here's the start of this one. Okay, so we need to fit in 13 amino acids. So we say this is 1, this is 2, this is 3, this is 4, I'm probably going to have too much space here, this is 5, this is 6, this is 7, this is 8, yes we are, this is 9, actually maybe not, this is 10, and this is 11, okay, perfect, this is 12, and this is 13, so you've got 13 amino acids in between those two cysteine residues that are on the two sides of the loop. Okay, so that's the structure then of a cis loop. Okay, and all of these nicotine-like receptor ligand-gated ion channels, or cis loop ligand-gated ion channels, are going to have this cis loop here, held together by two cysteine residues with 13 amino acids separating those two cysteine residues. Then after this, what will happen is the polypeptide will span the membrane once, it'll span the membrane a second time, then a third time. You'll have a large intracellular loop between the third and the fourth membrane spanning alpha helice, and then here is the carboxylic acid terminus. Okay, so let's do a bit of terminology here then. Okay, so firstly we'll name up the membrane spanning alpha helices coloured in pink here. Okay, so the first membrane spanning alpha helix is called M1. Okay, the second one is then called M2, the third one is called M3, and the fourth one is then called M4. Okay, right. Then the loop in between M3 and M4, I gave that a name as well. That's quite important. This is the site for phosphorylation of these cis loop ligand-gated ion channels. Okay, so they often get regulated by additions onto this loop. Okay, and this is known as the intracellular loop of the subunit of a ligand-gated ion channel. And for short, the intracellular loop can be abbreviated to the IC for intracellular and then L for loop. Okay, right. So that's a subunit of a cis loop ligand-gated ion channel. Okay, right. So this is a massive, great family of receptors with a number of important families of receptors within it. Okay, so what are the important families of receptors that are in this superfamily of ligand-gated ion channels? Well, probably the most famous is the nicotinic acetylcholine receptors. Okay, and these are the ones after which the entire superfamily is named. So these are the nicotinic, and then, so the little n is for nicotinic, the ACH is for acetyl, okay, so the A is for acetyl, and then the CH is for choline, okay, and then the R is then for receptor. Okay, so there is not just one nicotinic acetylcholine receptor, instead there is a whole family, there's a huge number of nicotinic acetylcholine receptors. Okay, and some of them have as their agonist, okay, as their exogenous agonist, nicotine. Now, not all nicotinic acetylcholine receptors respond to nicotine, and you should be very glad of that if you do indeed smoke. Because there are nicotinic acetylcholine receptors on your skeletal muscle cells, and it is these which, when activated, cause your skeletal muscle to contract. Okay, if nicotine activated those nicotinic acetylcholine receptors which are on the skeletal muscle, then every time you smoked a cigarette, you would have uncontrollable muscle contractions. Okay, so you'd have convulsions basically, and it would most likely be fatal because it would cause convulsions of the diaphragm, and then you'd probably not be able to breathe. Okay, so you should be very thankful that nicotine is not an agonist for all nicotinic acetylcholine receptors. It's an agonist for a number of them that are in the brain, and that's how it exerts its addictive properties. Okay, right. So the nicotinic acetylcholine receptors are a family of receptors that are in this superfamily of cis-loop ligand-gated ion channels. So they all consist of 5 separate subunits, and all of the subunits have the same basic structure here. Okay, right. The next big family is the star of the show, basically, the topic of this video. The GABA-A receptors are all cis-loop ligand-gated ion channels, or nicotine receptor like ligand-gated ion channels. Okay, so these are the star of the shows. These are the stars of the show. We'll come back to them in a moment. Okay. Other examples of cis-loop ligand-gated ion channels in humans are the 5-HT3 receptors. So there are a huge number of receptors for 5-HT, which is also called serotonin. Most of them are G-protein coupled receptors, but there is one family of serotonin receptors, or 5-HT receptors, which are ligand-gated ion channels, specifically within this family of cis-loop ligand-gated ion channels, and they are the 5-HT3 receptors. Okay, and again, there is not just one of them. Okay, there are multiple 5-HT3 receptors. Okay, then finally, the other main example within humans are the glycine receptors. Okay, so glycine is a neurotransmitter which is used in the spinal cord, and there it acts as an inhibitory neurotransmitter. It makes neurons less likely to fire action potentials. Okay, and it acts through glycine receptors, which are cis-loop ligand-gated ion channels. They're all pentameric, and the subunits all have this basic membrane-spanning topology that we've seen here. Okay, right. So, that now puts the GABA-A receptors in the broader context of ligand-gated ion channels. What we're now going to do is, in the next video, we are going to focus in completely on the GABA-A receptors. Okay, we're going to look at all the different types of subunits for GABA-A receptors, because unfortunately there is not just one GABA-A receptor subunit. That would be lovely if there was just one GABA-A receptor subunit. It looked like this. You put 5 of them together to get a GABA-A receptor, bingo. It's not like that, unfortunately. Instead, there are 18 different GABA-A receptor subunits. Okay, and we'll go through all of the different GABA-A receptor subunits in the next video. Actually, I think it might be 19. Sorry, there's more than 18. It's worse. There's 19 GABA-A receptor subunits. Okay, right, but we'll go through those in the next video.